I'm Natalie, a lab technician in bioarchaeology at the University of Auckland. My main area of expertise is in analysing ancient DNA, usually from archaeological animal bones. Archaeologists often find animal bones at sites where humans once lived. These bones could have come from farm animals or animals that were hunted for food, from pets like cats or dogs, or just from pest species like rats. Bones contain cells called osteoblasts, osteoclasts, and osteocytes, and these cells all contain DNA. Due to the inorganic mineral part of the bones preserve in the ground better than the rest of an animal's body tissues. This means that DNA can stay preserved too, sometimes for thousands of years. All DNA is made up of the same 4 basic molecules, or nucleotides, and the order of these molecules is called the DNA sequence. It's this sequence that determines the types of proteins that are made by each cell in the body. Animals of the same species or population have DNA sequences that are more similar than animals of a different species or population. This means that by analysing the DNA from archaeological bone samples, we can tell which animal species or population that bone belonged to. When bones are found at archaeological sites, they're often just fragments of bones that have been damaged and broken into small pieces over time. By carrying out DNA analyses, we can figure out which species of animal these bone fragments belong to. Also, sometimes archaeologists find tools made out of bone, like fish hooks or needles or arrow points. So DNA analysis is really useful for identifying which animal bones were used for this. As well as just identifying a species of animal, we can sometimes identify where animals first came from, how they evolved, and how they got to where they are today, based on similarities and differences in DNA between populations from different locations and at different times. For example, the first domestic sheep came from Mesopotamia in Western Asia around 12,000 years ago. The first domestic cats were bred from Near Eastern and Egyptian populations of the African wildcat around 10,000 years ago, and horse riding originated in Central Asia about 6,000 years ago. Understanding the way these species evolved and the way humans interacted with animals in the past can give us a real insight into the lives of our ancestors, as well as an insight on our relationships with animals today. So when I'm working in the lab as a technician, I help researchers with their ancient DNA projects. We start out with bone samples like the ones on the left, which have been collected by archaeologists in the field. We then take a very small subsample of less than 0.1 grams and we grind that into a fine powder. The next step after this is to extract the DNA from that bone powder, and this first involves demineralizing the bone, then using an enzyme to lyse or break open the cells. We then separate the proteins from the DNA, we precipitate and clean the DNA, and then we rehydrate that DNA in a buffer solution ready for further analysis. This analysis isn't carried out just by looking through a microscope. The DNA is way too small for that. Instead, the most common method used once we've got our extracted DNA is a process called PCR. Through PCR, we can make thousands of copies of the section of DNA that we're interested in. And during this, what we can do is add a dye to each of the 4 molecules called nucleotides, and then use a DNA sequencer to view those coloured dyes on each molecule. At the bottom here is an example of what DNA sequence data looks like at the end of the lab process. From this point, we can then read the DNA sequences and analyse the data. For example, we can create an alignment like this one to compare our sequences to other sequences in our set, or ones that are already available from other lab groups. And we can use this alignment to build a phylogenetic tree, like the one on the left. We can identify the species, as I mentioned earlier, or investigate a range of other research questions on the animal bones, helping us to better understand past relationships between humans and animals.